Move on to the next talk. We have Darshit Patel, who is CEO and co-founder of Decode Age. It's a company that works on microbiome sciences. Over to you, Darshit. Thank you.
Yeah, hi folks, how are you all doing today? So, we'll get a little bit of exercise in, and the exercise is very simple. I'll ask you two questions, and if you agree with me, just raise your hands. That's the exercise. So, the first is how many of you are actually excited about microbiome being a part of longevity diagnostics? Yeah, lots of hands, I was expecting that. And how many of you are very, very excited about use of ML and AI in microbiome? Right. Now, while you park that thought aside in your head, let me ask you a question. Imagine this, you have gone to two hospitals and both of them are using a different thermometer scale, right? One of them says that you are hypothermic, the other one says that you have a fever. Would you think this is absurd in any normal scenario? Well, this is what's happening with microbiome as of today, and because the reproducibility is an issue, we'll talk more about it as we go on. So there was this very interesting group in Italy which did some work, and they came up with a consensus paper that microbiome technologies lack reproducibility. Every time you send your sample to someone, they'll give you a drastically different result. And there's a very famous magazine called The Microbiologist. Linda Stewart said that this is the wild west of microbiome diagnostics. So I'm a curious person. I went on to see, did cowboys in Wild West have any rules? Well, they had. If it ain't true, don't say it. If it ain't yours, don't take it. If it ain't right, don't do it. Well, I agree with the cowboys here a little bit, and especially I wanted to bring this into the microbiome technologies, right? So that's how my journey started. Back in 2020, we started with the microbiome project, and we realized that there were some standing issues with the technology which is making it completely unreliable. And the first is the reproducibility itself. Its microbiome technologies are notoriously hard to reproduce, to be honest. That means that if I'm sending a sample to you today and I'm sending the sample to you tomorrow, it will have drastically different results. And this is not just because you are biologically different in a day, but also because of the technicalities of it. A group called Microbiome Quantitative Check QC, yeah, Microbiome QC group, they did a large scale study. They sent the samples to different labs, and this is what they finally found out. They found out that when you're using this different extraction, different sequencer, which is your Illumina, Oxford Nanopore, even variants of that, you get some kind of variation associated with that. Now usually you'll see that here, this is the biological variation, but these are caused by specimen extraction and sequencer. Although this looks very low, right? When you have large amounts of data, this will automatically magnify the noise in your data. This is what we call the technical noise. My most interesting realization was out here. You see, this is the biological, the pinkish thing off, and the purple thing, which is the kind of bioinformatics procedures used to analyze this data. This brings us to a very important question, and which is that of digital contamination. We have always heard about labs contaminating samples, have we not? Yeah. There's something called digital contamination, and many studies have been published which says that the reference database that you are using, be it your NCBIs and the other, when you are mapping your data to it, the sequences stored in this reference database itself are very much contaminated. So whatever you are predicting, it's pretty much wrong. And a very high profile Nature study coming from a very important microbiome lab in the world proved for a certain time that there are tumor specific tissues, there are tumor tissues having its own resident microbiome, and they proved that different cancers could be identified based on that. Well, their optimism was short lived because Salzburg lab then came out with a paper, and they said that pretty much everything that they predicted were human sequences and not really bacterial sequences, and it led to retraction of this particular paper. And you imagine a startup was being built out of it, it had to shut down purely because of that. And this is one of the graphs from this particular study which challenged the original study. You see, this is the original study. The number of bacteria they found were close to 500,000, and pretty much so many genera of bacteria was found. Then they reanalyzed, adjusting the reference database, and suddenly the top hit was nearly 270 bacterial reads only. Now this was the problem, and this is why the startup had to shut down. Also the paper got retracted. Now you might be arguing that this is because cancer tissues are very low biomass samples, there's more error prone and everything. But let's look at gut microbiome data, right? Very interesting stuff. Someone built a tool specifically to check for fungal species in your human gut microbiome. And they thought they did a fantastic job. They developed an entire database with fungal taxons. They were so happy. Everything was predicting just right. And then someone else came in. What they did, they changed their reference database with the original reference database. And suddenly, if I may ask one of you, do you think that your gut would harbor crocodiles, snakes, and salamanders? Frogs, salamanders, turtles, crocodiles, snakes, all found in human stool. Purely and purely because of digital contamination, which no one cares to talk about honestly, be it academia or industry, and which is why I really wanted to highlight this. And now, the term we are all been speaking since last 1.5 days, right? AI. AI. Who all is excited about AI? Yeah, everyone is excited about AI. So what happens is that there are multiple disease prediction algorithms being made by in microbiome, and you'll see a lot of direct to consumer testing companies going around and giving you disease prediction based on your microbiome profile. Another important retraction is on the charts, right? Because someone made an obesity prediction algorithm from microbiome data, and then again a different group of researchers analyzed the same data, and they figured out that there was one confounder every microbiome study has missed out of, which is microbial load. And then they took all the existing data sets. You remember the famous study for the Hasda hunters and the unique microbiome they have compared to the urban people? Well, they took the same data set and then they tried to reanalyze it, keeping microbial load as a confounder, and readjusted all the models, and suddenly all these important names you see, Faecalibacterium prausnitzii, Bacteroides, Clostridium, all of them lost 5 disease associations each and some even more. That means pretty much all the algorithms that we know for disease prediction are completely wrong. So do not believe them. So you are wondering now that what is this industry guy doing here, hurting his own business model by talking about the problems that microbiome industry is facing while himself doing microbiome test. Well, I'm also a founder as much as I'm a science enthusiast. So unless and until I'm solving for it, I'm not going to highlight that problem. And that's what we have been doing as well. So this is a plot from 2021. We took the same biological samples and replicated them. That means we broke it down, we analyzed it at different time points, different runs, everything. And we saw that the same sample, when different sequencers were used, were biologically distinct. You see, it's scattered all over again, right? That means that every sample is different although coming from the same person. Now, how could you trust this data? This was the first roadblock we hit. Second we hit was using these standardized mechanisms and benchmarks of microbiome testing. We followed the same procedures, analyzed the same data again, and we saw that the precision or the confidence that I'm having in predicting a particular species out of your gut is 75% only, and that too at a very high confidence rate. If I do it in default, which is the general practice everywhere, the confidence is 0.51%. That means if you are having a default pipeline and if you are predicting your microbiome species, there's 0.5% chance that that is right. And that is our first roadblock that we hit. Now, honestly, when I started Decode Age, I wanted to build something fancy, a crazy AI, which will tell me what's microbiome and longevity connection. How do I solve it? So exciting, right? And then we saw all this, and then we hit a roadblock, right? Suddenly the startup's mission from being microbiome and longevity company went down to fixing lab protocols, doing boring fundamental work, right? There were two options for me. Either I could have just fooled everyone and keep going about it, or I could have sat down and fixed it. But fixing it means taking 2 years, 3 years. So we did. We had to fix it. And that's how we started. And now the next series of experiments, we fixed the lab protocols, we fixed DNA extraction, how the samples are connected, how they are stored, how they are analyzed, everything. And the same reproducibility chart the same sample split across different replicates, analyzed after 1 year of storage in different sequencers in different time points, and see how tightly they are clustering the similar type of samples. Do you see the scatteredness anymore? So this is what we exactly did with lab protocols to first fix how we are even looking at microbiome. And then we went on to look at a little bit more resolution, because a PCO is not going to tell me everything. So now there are three important pieces in microbiome: the genera, the phylum, and the species themselves, right? So can I find reproducibility across all of them? Well, this is the same sample sequenced thrice, and you see most of the species are represented in a very similar amount. Same with the genera, and same with the phylum. So we now are quite confident that what we are predicting is reproducible, is reliable and robust, because we also changed our precision to about 100%. I agree, when I change precision to about 100%, I might be losing out on some species, but it's important that what I'm predicting, I'm very sure about it rather than what I'm losing out. And for things that we are losing out, we have already started working along those lines as well. So now this is essentially the story of Decode Age and the Decode biome that comes with it. Our job was not this though. Since last 2 years we took a detour, right? And now we have to get back to clinical research, something that can have impact in overall aging and longevity research. And that's when we again started right from the scratch, taking up clinical studies again. And thanks to some important collaborators here, I think with your trust, we could do a lot of these cohorts now. We work with Dr. Vishal for one of the cancer therapeutic prediction, which is essentially what we are trying to do, is we are trying to tell if a microbiome profile could actually tell which therapeutic a person is going to respond to in cancer. That's one of the cohorts we are working with. We are working with Dr. Reddy's to find inflammatory signals coming out from gut in IBS patients and even in gut-brain axis. And a very interesting study we did in Zoograma. I think pretty much everyone who is doing microbiome test here is looking for a personalized food recommendation. Like I previously said, there's nothing like that unless you go to a certain extent of personalization. So we did this really interesting study in January, and I don't know, most of you are not aware of Zuzulu cities or something. It's a pop-up city, 6-week people come together, right? They live together, they co-build things together, and they're pretty much in the same area. And we know that microbiome intervention studies have been plagued by environmental variables, technical variables, and everything else that comes with a person and their metadata. We've solved for technical variables already. In Zoograma, what we did was 6 weeks people were living in the same place. We allowed 2 weeks to settle their microbiome. It's back? Yeah. We allowed 2 weeks to settle their microbiome. Then we did a day 0 collection of their microbiome samples. And then for the next 3 weeks, believe this or not, everyone ate the same food at the same time, lived at the same place, and followed at a particular lifestyle that they were following. And now we are looking for signals around that side. And finally, we have a spin-out collaboration with IISc and Decode Age, where we are particularly working on a microbiome therapeutics. Again, more precision in nature, and we are testing out first with cancer. So that's pretty much me. Happy to take in any questions. I saved a lot of time for the next speaker.
Thank you, Darshit, for that beautiful talk on the errors that are happening in analysis. Quick questions, 2 quick questions. Yes, up there.
Are you doing anything for the autoimmune disorders? Lot of is there on the internet that did you leaky gut and microbe change in the gut, the autoimmune diseases are there which have no cure practically.
Yeah, I mean, see, there's lots of data on microbiome and autoimmunity, but like I just said, can we trust most of this data? Probably not. Secondly, if I'm to work in India, I would obviously like to collaborate with clinicians who are working in autoimmune conditions. Let's first build a cohort. Let's first see what's understanding we are getting out of it, and then we actually move on and go into clinical practice. And I think this is a point highlighted by Matt and other speakers earlier as well, that we shouldn't rush into clinical practice unless we are very sure of something. So I'm happy to collaborate. We are already collaborating with a lot of partners working on different conditions, where our job is to make sure that the microbiome testing that you are using is quite precise and consistent. And that's the goal, yeah.
Question there.
Thank you, Darshit. This is on your right, Tavpatesh
Hi.
here. Thanks for highlighting the reproducibility issue. Great work. My question is towards the use of precision as one of these indicators. We know that precision is heavily dependent upon prevalence as well. And microbiome being a mixture, if we are looking at precision as an optimizer, we are probably only looking at the highly prevalent species and perhaps missing out on the ones that may make a difference between health and disease.
Yeah.
So, do you see a trade off there, or are you also looking at that as a potential area of improvement and using other markers other than precision? Because if you actually chose a highly prevalent, most prevalent species, I would assume that your clusters would be by design they would be tighter than if you included something which is more variable, less prevalent. What's your thoughts on that?
No, I think I absolutely agree with that. And this is a problem I'm tackling in the lab as well, right? Because I know that 100% precision and losing out on certain species, that means I'll be losing out on rare species, right? So usually then we see what perspective we are looking at microbiome from. If I'm going into clinical practice, I should be looking at a higher precision. But if I'm looking for new signals in a new research study, I might compromise a little bit on the precision and see if my recall is high so that I'm actually predicting everything that is there in the gut to start with. So I think to answer that question, it's more of what the objective of the work that you are doing so that you can actually figure out what customization you want to your pipeline and data analytics.
Thank you. Thank you, Darshit. And we'll stop on that cautionary note. Thank you very much for your time.
Thank you.